Welcome, everybody. It's lovely to see you all there. Just so you know, the webinar is being recorded, in case you weren't aware, and it's brought to you by the Research and Development Consortium Ecopath and by the Marine Board. And now let's see if I can get my screen to, there we go. We have people coming in. So some house rules: no heckling from the back seats, please. Please ask your questions by raising your hand. If you have any technical support, put it in chat and maybe I'll be able to resolve it, but probably not. And you will be able to re-watch this on YouTube, either through the Marine Board or through the Ecopath YouTube channel. And today, oh yes, hold on. This is a seminar series happening every 2 months or so, usually on the last Tuesday of the month at 6 o'clock Eastern, European Central Time or summertime. So we have 2 dates, hopefully for the 25th of July and the 26th of September. And I think we have somebody planned for the 25th of July, if I'm not sure. Sometime in July anyway. And today we have Daniel Vilas here, who is going to talk to us about red tides, evaluating red tide effects. So thank you very much for being here, and I will stop sharing and Daniel can entertain us.
Hello, everybody. Let me share first my screen. Can you see my screen? Yeah.
Yes, we can. Thanks.
Okay. So thanks for the introduction, Sheila. Today I will talk about red tide effects, a work that I've been doing for the last years at the University of Florida. This work is part of a big ecosystem modeling project in the Gulf of Mexico that was funded by the NOAA Restore Science Program. And this project was led by David Chagaris. And so let's move into ecosystem modeling. So harmful algal blooms, also known as red tide, consist of the massive proliferation of micro and macro algae species, and those proliferations cause toxicity in the waters. So this is leading to a growing concern in terms of the impacts that cause in the system. And if we take a look on the bottom left maps here on the slide, we see the increase in those red dots represents red tide events. We see how difference between the picture from 1970 to 2017, how these red tide events grow in terms of frequency, but also in terms of severity. And this is leading to getting the attention of media, as we see this image from a weather forecast of Florida. Also some warning signs because of the effects that those events cause on the people swimming in those areas or visiting those areas, causing irritation on the skin and eyes. But also, apart from the human health impacts, it also impacts the ecology of the system and also the economic, getting to get some loss on revenue for the tourists, but also in terms of harvest and aquaculture. Here we have in the US was estimated the impact on commercial harvest fish and shellfish was 5.6 billion 2018, and in 2017 for aquaculture production was 1.5 billion. Something that we have to keep in mind as a size of the impact. Those events are usually are caused by Carenia brevis, is one of the most common species. These species are a dinoflagellate that segregate this toxin called brevotoxin that affects the nervous system. And those species are present in background concentration on the regions that are on marine system, coastal systems. And then some factors that are not well understood provoke the massive proliferation of those populations. And the factors may be different. There have been studies that suggest that the agricultural runoff are one of the main causes for leading to those impacts. Others suggest the position of Sahara dust, and others suggest some upwelling or current events in the region. Those factors have led to the nature of those events that are pretty localized in terms of space and time, with a very short duration, which leads to a very unpredictable negative effects, something that is makes a great challenge for management. In terms of ecological impacts, several studies reveal that there are ecosystem impacts, also food web impacts causing trophic cascades through the food web. It may also affect the behavior. It may potentially cause the shift on the distribution of a species, and also increase the mortality or reduce the catches, as was reported in other modeling studies. To deal with these impacts, ecosystem management offers a holistic approach in which we can consider environmental and other connections in the system, trying to achieve the sustainability of the system, and considering the structure and the function of the system. Among different ecosystem management levels that we have differentiate in that diagram on the top right, we have the ecosystem-based fisheries management. That is the one that we will focus on this talk today. That focus on fishery system within an ecosystem, as well as trophic interactions, habitat associations, and climate variability. In 2017, the NOAA released a roadmap for the implementation of EBFM in the US, and ecosystem models have been used to advance to EBFM. For example, we have the Ecopath with Ecosim that maybe sounds familiar to most of you, and also the Atlantis model. But we need to consider that there are some fisheries management plans that require a special framework because of the nature of the issue or what we want to implement, where the spatial temporal scope of the problem is very important. And to do that, we have those spatial temporal ecosystem models that comes to the bottom. At the bottom right, we have these numerous ecosystem modeling tools and what they are used for. And we will focus on the last row, the end-to-end ecosystem models. And to apply them for management, we have to acknowledge that those models need to pass through sensitivity and uncertainty assessment, but also we need to calibrate and validate those models and validate those predictions. This talk will focus on the West Florida Shelf because it's a region that it's regularly impacted by those episodic red tide events and causing severe impacts in the region. We have, for example, this map on the bottom right that represents those cell concentrations of Carenia brevis, and the purple ones are the severe, the higher concentrations. So in late summer, we have those episodic events, very localized and pretty short in duration. The West Florida Shelf is a region on the northeast Gulf of Mexico. It covers 180,000 square kilometers with a broad shelf, seasonality on temperature, salinity. It hosts highly diverse fish communities, especially reef fish species and habitats. And also it fosters available commercial and recreational fisheries. And in this region also, there are many ecosystem pressures that affects the system at different temporal and spatial scales, such as overfishing, red tide, hurricanes. We have also oil spills at some point, invasive species, so many pressures. And in the West Florida Shelf, there have been some work done with ecosystem modeling, suggesting that red tides may affect the community structure and may cause cascading effects in the region. But we have to recognize that those problems, given the nature of those problems, there are some management plans that require this spatial temporal framework. And spatial temporal ecosystem models have been used already to investigate management plans. For example, the work that Kim Dematzer have been done in the Delta Mississippi River with the restoration, different restoration scenarios. And so spatial temporal ecosystem models can fully quantify those spatial temporal impacts. And to do that, we have to take into account the lethal as the mortality effect of those red tides, but also the sublethal effects of those stressors that can represent the avoidance to those impacted regions. And the localized and the fact of getting those quantify impacts of the red tides remains two of the great challenge for the assessment of marine resources in this region. Red tide have been considered in recent stock assessment in the Gulf of Mexico through this what it's called CEDAR. And for those reef fish species here on the bottom left, the gag grouper and the red grouper have been included on the last stock assessment red tide information. For in those stock assessment, what was done consists on creating a pseudo fishing fleet in which the mortality caused by this fishing fleet is attributed to the red tides and is activated during the severe red tide years, meaning that it is estimating this fishing mortality, what is not fishing mortality, it's other natural mortality for predefined years. And to have an idea, in 2005, the red tide mortality rate for gag grouper was estimated between 0.99 and 0.35. And here we have some plots, how those plots represents different projections and their different red tide conditions. So how will change the yield and the depletion state of the population when consider different red tide events in terms of severity. How severe were those red tides in future years? So the objective of that study was to improve the parameterization process and increase the robustness of spatial temporal ecosystem models contributing to EBFM, help to incorporate those spatial temporal stressors into ecosystem models, and tease apart those lethal, sublethal red tide effects, and also the indirect food web red tide effects. All that will be with the objective to provide a comprehensive assessment on the quantification of red tides for the improvement of the management of some populations in the Gulf of Mexico. To do that, we use the Ecopath with Ecosim software. I'm going to briefly pass over here. We have the Ecopath, that is the static version of the system. Species and functional groups are represented and connected, represented through the interchange of organic matter, matter and energy. And these models are ruled by two main equations here, the consumption, the first line, and the second line, the production. Then we have the Ecosim, that is the dynamic version of the model in which we drive this model over time, projecting those observations, fitting those observations, and getting through those differential equations on biomass that are represented here on the bottom. And on those models, we incorporate, there is an incorporation of the vulnerability parameters that regulates this trophic interactions between species. And then we have the Ecospace, that is a spatial version of the model. So bringing that temporal to a spatial scope in which the system is represented by a base map of grid cells in which species move around depending on the quality of those cells. The Ecospace model incorporates this habitat capacity model that is one of the, it computes an index of quality for each of the cells in each of the time simulation and each of the functional groups in the model, that it's computed by multiplying the effect of each environmental factor included in the system. And the species move around in terms of the quality of the cell that it also established by also the level of prey for that group. It also includes the spatial temporal framework that allows to incorporate the spatial temporal information into the system and allows to a better representation of the system. And also we use the individual based model that allows that groups are moving into groups of packets of individuals, which allow a better representation on the spatial predictions. The West Florida Shelf model is an updated version of Chagari's 2013. It runs 36 years at monthly step. It includes 17 fleets and 83 functional groups. It includes depth, rugosity, temperature, salinity, and primary production and red tide as environmental factors. It also includes response functions from fitted binomial general additive models from fishery independent data. And the main components of that model that were used to tweak and tune the model were the response functions that are the response of each individual in terms of fitness to an environmental variable, the vulnerability, that is the rate of exchange of a prey from vulnerable state to invulnerable state, and the dispersal rates, that is the ability of those species to move around that base map. We start, once we build the model, we start an stability process. This stability was is a stepwise process in which we start building the environmental complexity in the model, going from the static version of the model in which there is no temporal environmental variables. And then in each step, we build that complexity. First, including the primary production, then including the temperature and salinity, then including the red tides to achieve a stable model. And I talk about the stable, meaning that we try to avoid exponential increase in terms of biomass for functional groups or collapse of some species. When we bring such amount of spatial temporal information into the model, we may find that there are some groups that are crashing or are increasing where there is no sense for such increase. And in each of those steps, building that complexity, we evaluate the root mean square error and the Euclidean distance by comparing the Ecospace prediction with the Ecosim predictions. In this case, our Ecosim predictions represents our true values. So we wanted our Ecospace that they approximates to our Ecosim predictions. Then after running this stability process, we ran a spatial validation in which we get the predictions from Ecospace, average all those spatial predictions and compare with the generalized additive models, the predicted probability of those GAMs. So we have maybe a good understanding of which groups maybe are not especially behaving properly. Then after that, the validation, we ran that calibration process or fitting. We did that. We follow the approach of trying to approximate our Ecospace prediction to our Ecosim values. And to do that, what we think is just an iterative process in which we consider all vulnerabilities and dispersal rates parameters available on this Ecospace model, and each iteration perform a parameter sensitivity in which we select the most sensitive parameters. Then once we got the most sensitive parameters, we run through multiple values for those parameters, and until we find the best value for that parameter, which leads us getting a lower percent of bias and lower, trying to minimize the likelihood between Ecosim and Ecospace. We did that until there was no more improvement on the model. So given we didn't get we couldn't get closer to our Ecosim observations, and this was 35 iterations, so 35 parameters. And it means more than 80,000 Ecospace simulation. And this was done through an Ecospace console that allows to run the Ecospace in through R, and using a computer station, we were able to run that process in parallel. So we can run that like faster than we should through the software. In terms of the incorporation of red tide effects, the first was developing those red tide severity maps. We got information from Karenia brevis, cell concentration from the fishery wildlife, the Florida Wildlife Commission, and this harmful algal bloom monitoring program. And what we did is first create those observations, but because of the nature of those sampling design, in which is very localized, because it's an opportunistic sampling, we need to set some boundaries. So what we did is get the satellite fluorescence map and create those polygons in order to clip those kriging from the concentration of Karenia brevis. And so we ensured that those predictions were not going farther from a bloom that was indicated by this satellite fluorescence map. And finally, we create that those maps here on the right side that are those red tide severity maps. And we have this in 2005 in September. We have this spike on red tide impact, well, red tide concentration. Apart from the severity, the red tide severity maps, we need the mortality response functions that represents the lethal red tide effects. And this is a novel feature in Ecospace that adds an additional natural mortality term into the equation. And we evaluate different mortality red tide response functions. These logistic functions differs in terms of sensitivity and also the slope. So we have different sensitivity levels because there were no information on the red tide response for a species. So the higher the cell concentration of Karenia brevis, the higher the proportion kill in that cell. And we did something similar to the sublethal red tide effects, in which we get we try to represent the avoidance. So the higher the concentration of that cell in concentration of Karenia brevis, the lower the ability of forage of that species in this cell. And similarly, we have to evaluate different sensitivity levels for that for red tides. In this case, the logistic function works contrary to the mortality case, in which the higher the concentration, the lower the proportion of foraging capacity. And this is one of the main outputs of the model, was the red tide mortality rate. On the left, just above the figure, we see this equation that it's dx represents the proportion, the concentration, the f is the inflection point, the b is the slope, and we got that proportion, in each group, cell, and month. And we got this instantaneous mortality rate, and it was a scale to the base other mortality, and incorporate here with the predation mortality, fishing mortality, to get all the lost in each cell. And the red tide loss, it was just multiplying the proportion, instantaneous proportion, with the biomass, and we get the annual by summing and averaging through all the months. So without all those sensitivity levels, and when applying or not applying those sublethal effects, and applying to only gag grouper, because we want to manage only gag grouper to all the food web in the ecosystem, we end up with 160 red tide scenarios, which represents more or less how the uncertainty in terms of sensitivity, but also in terms of what we want to evaluate. Well, we want to evaluate how the sublethal effects affect to the system or other ecosystem indicators. So we have these different indicators to evaluate different levels of the system. We have the ecosystem red tide impacts that were evaluated through biomass and catch indicators. We compare, you will see that we differentiate between using or not using those sublethal effects, something that we did also for the community red tide impacts, where we compute a biomass indicators for taxonomic and trade groups, and then the population impacts that were based on the gag grouper population and were filtered in terms of the red tide lost in 2005 that was estimated in 0.77 for the last gag grouper assessment. And also lowering the root mean square error, trying to decrease the observations with the predictions. And we differentiate when applying red tide effects to the whole food web system or just to the gag grouper population. And so we could differentiate if there is any indirect food web red tide effects. And we have this table here where we see the ecosystem community here, those indicators, and the population is just using all those but filter as I mentioned. And finally, there are those, we need to evaluate the effects of red tides validated by comparing observed values from fishery independent data with the predicted biomass from Ecospace. And we did that from inside, by comparing inside, outside the red tide event, but also before, after red tide events. It depends on how was the data that was available for that year or that month. Here are the representation of the Ecospace stability, the root mean square error result going from the static on the top row to the last row is the fully dynamic. And this just, I put that in that slide just to visualize how the functional groups evolve as you build that complexity in terms of spatial temporal information. There are some also, there are some, it helps to visualize which groups we want to pay more attention, where there are more difference. So we have this more dark red. And similar to the Ecospace validation. Here we have each row represents a indicator in which it's comparing the Ecosim the Ecospace long-term prediction with the GAM response functions. And we see different values. The one with the X, this cross represents the ones that have not good value for each indicator, meaning that we need to pay attention to how the spatial representation of that species. And moving to the ecosystem indicators, the ecosystem indicators reflect degradation and fisheries impacts in the region. We have these 4 indicators plot that represents the lines. The black line represents no red tide effects, so ecospace without red tide effects. The yellow represents the ecospace prediction when including just lethal effects, and the blue line represents when including lethal as well as sublethal effects. And we see that there, in 2005, especially, we have this harvest biomass loss on harvest biomass, and increasing biomass loss due to red tide. So revealing that the 2005 was the most severe year in terms of red tide impacts. We have some kind of differentiation at some points for the blue line, given those spikes that could represent some kind of compensatory responses due to those sublethal effects in terms of avoidance or in terms of competition release at some point. If I remember well, specifically, there are some that reflects are represented by crabs, increasing on crabs, especially those harvest biomass. But there are those spikes here are represented by other groups that are not well modeled. For example, the jellyfish, that are groups that sometimes cause problems because of this their PB and QB ratio. And we have a similar picture for the community impacts. We have this decrease in 2005 for most of the groups, but they behave differently in terms of recovering from that 2005 red tide year. The differences between groups are attributed to difference in production ratio or even cascading effects. There are some groups with PB ratio, which probably are not allowing them to recover, others that recover faster, or others that are affected less because of the overlap between their species their distribution and the red tide impacted area. We have also some differentiations when applying or not applying those sublethal effects. We see that there in some cases we have a higher relative biomass when using those sublethal red tide effects, meaning that maybe it's helping some of those groups to avoid those impacted regions and swim away from those areas, since they are losing the foraging capacity. Moving to the population impacts, these plots here represents the 160 scenarios, but after filtering them, so we have these different facets that represent when a red tide were applied to gag grouper, only using lethal effects or sublethal effects, or to all consumers, so all consumers in the food web, and those different sensitivity levels on the x-axis, on the x facets. And then we have the ones that fall into this gray rectangle. There are what we call good red tide scenarios in terms of they approximate to what we see for the gag grouper population in the stock assessment, because they lower the root mean square error, or the differences, the difference on the natural mortality for 2005 was low. And this gives you an idea of where are our sensitivity approximate. So we have that maybe it includes sublethal effects, it's better because we got more numbers, and also we have this this low sensitivity doesn't work for the natural mortality, but for the root mean square error it works better than the other ones. And here we have the red tide mortality rate for the gag grouper. We see higher mortality for the younger stanzas. Those were the stanzas of gag grouper included in the food web, even much higher than the older individuals, and that's because of the spatial overlap between the red tide events and the distribution of those stanzas. And we have the same thing here with the 2005 as the most severe year, and other years following that doesn't get much lower mortality, but they still got some mortality. 2012, 2005, and here we have like a kind of comparison between the red tide severity in 2005 and the gag-free mortality. How are this group responding to those events here in September? This mortality here. And so what we did also was create predict this biomass vulnerability gag grouper index. This plot here represents observations from fishery independent data. We have this yellow line representing the prediction from stock assessment. Then the black line that represents the ecospace without the red tide effects. The red line represents ecospace with direct red tide effects, meaning that the red line only represents when red tide effects were applied to just gag grouper. And then the green line represents the ecospace predictions when applying red tide effects to the whole community in the system. So if we see that there are this decrease in 2005, that sometimes is not captured by the observation. And this is mainly due that those surveys are not focused on capturing the mortality from red tide. So they have another objective. And another thing that we want to take a look is this H0 survey that represents the recruitment on these populations. We have this kind of different, a longer and kind of higher recruitment when using those red tide effects to the whole community. And after the 2005 event, where there was a peak in mortality, and this is may suggest that there are some kind of trophic-driven compensatory response, likely to the competition release from when applying the mortality to the whole food web, you are releasing from competition to other functional groups. So while single stock assessment models rely on fishing, mortality, natural mortality, and recruitment deviation, this Ecospace model really rely on fishing, food web, and red tide effects. And in terms of the validation of those red tide effects, here we have a pair of maps with a plot. The plot on the left for each of those blocks of plots, the plot on the map on the left represents when no harmful algal bloom. So before or outside for the case of the plot number D as Denmark. And the map on the right or on the center of the block represents inside or after this red tide event. We see this change on biomass here, for example. And the plot represents how we observe these red tide effects through survey, through fishery independent survey, and how we observe them, well, how we predicted in Ecospace. The blue point represents when no harmful algal bloom event was happening, so before or outside, and the red represents when harmful algal bloom was present or just after this red tide event. And for example, here we see that the Ecospace over predict these red tide effects compared to the fishery independent data. But in 2014, we see the opposite. So our Ecospace prediction underestimate the effects of red tide. We have a decrease in biomass here that was not captured by Ecospace. And then from the 2015, we see more or less similar. So we were more or less approaching that real values of the red tide effects. And so in conclusion, the West Florida Shelf Ecospatial model was able to quantify red tide effects. Severe red tide effects occur on the West Florida Shelf at ecosystem, community, and population level in terms of biomass, catch, and productivity, especially in 2005. Sublethal and indirect food web red tide effects trigger trophic compensatory responses. The Ecospace model calibration helps to increase the reliability of those models. We have to acknowledge that those models sometimes can be doubting because of the amount of information that comes into those models. So given that previous process through the building of that calibration, it increased the applicability. And the fact is that we want to drive that process to operationability, driving the management of those red tides to a more effective and proactive way to do it. And another important thing was to understand the dynamics and effects of those red tides. We need more information on how species react to those events, and how can we improve the parameterization of those models. And we got one of our objectives that was including this red tide mortality rate index to inform the single stock assessment, or at least to include it in alternative run for the last gag grouper stock assessment that may help to contribute to the EBFM. And we build this R Shiny app that we think that it may help to represent the uncertainty of those where we do to visualize that uncertainty and communicate that uncertainty in terms of the sensitivity to red tide events. And some future steps on that model is that those models may push to create more samples and monitoring program to get data from those stressors. We want to manage some stressors, but sometimes we lack of that data. It was difficult to find before, after, or inside outside data to evaluate how our model represents the red tide impacts. It may help also to evaluate alternative stressors, such as oil spills or other kind of stressors that may apply. It could be used also, the West Florida Shelf Ecospace model, to evaluate unprecedented red tide scenarios, and also inform other stock assessment, such as the red grouper stock assessment. We can quantify this red tide mortality rate and apply to a single species stock assessment model. It also helped to create new opportunities for those spatial temporal ecosystem models in terms of using as operating model to evaluate different kinds of sampling design. And finally, this plots represents the work that I've been still working on, and consists on getting that, using that ecosystem model, spatial temporal ecosystem model, as operating model to generate that red tide information, and input that information into a stock synthesis model to evaluate the different ways to incorporate that information into the model, and how these affect the the outputs of the model. So do we want to incorporate that information as an environmental variable, as a block of mortalities, as a bycatch that in the way that it's been doing currently on the stock assessment process? What is our best option? And with that, I would like to finish by acknowledging the help of many colleagues here, and especially to my former advisor, David Chagaris, and the fund from the NOAA Restore Science program. And with that, I will take any questions or comments or thoughts.
Thank you, Daniel. Ah, there we go. Hi, Carl.
Yeah, great talk. I was living in Sarasota, just south of Tampa, during I think it was a 2002 red tide event, and it was horrible. It made people sick, and it killed virtually everything in the bays along the shore. It didn't have a larger offshore effect, but I knew a bunch of commercial fishermen. And talking to those fishermen, they had really mixed feelings about the red tides. They said that historically what they did when a big red tide hit that area around Tampa was to move north up into the panhandle for a year or two and live up there and fish up there. But then they expected when they came back to the Tampa area to have incredibly good inshore fishing for crabs, mullet, small pelagics and stuff like that. And I saw that there was a huge bloom in juvenile catfishes and small pelagics and so on in those bays in the year after the red tide. So it released a really strong trophic response in the system, a lot stronger than your model indicates. Do you have any idea why maybe the model didn't capture the really strong response that those fishermen expected to see? It was juvenile fishes and juvenile crabs and things like that that really exploded, probably because of removing virtually all the predators from those inshore areas by the red tide.
So yeah, first of all, I think that there is like this kind of thought about depending on who is where are you working, is like, well, the worst red tide was in 2012, or no, the worst was in 2019. Depends on where are you living or where are you working at, right? And the other thing is that, yeah, I was expecting I agree with you that I was expecting the match trophic response, and probably that's maybe due to the fact that we were evaluating different kinds of severity, and probably we don't see as much response of some groups because there are some red tide sensitivities that were like lower in the importance of other ones that we were taking into account. So I think that maybe if we will have a better understanding of how the species react to those events, we were able to better quantify those trophic response and get it a much approximate or higher response of several groups.
We tried to, Dave and I did. We couldn't get any of the Ecosim models to show as strong a response as I observed and that these fishermen talked about. Good luck on this.
Thank you, Carl.
Christy, maybe you have some help.
Yeah, I was gonna say, I think that's can you hear me okay?
Yeah.
Okay, good. Yeah, I like Carl's question. And so one thing that maybe I missed is what data set did you use to calibrate the models? I mean, the FIM data for the state of Florida is really good in this region. Were you using, I mean, because I could see, did you evaluate the responses just over time series and things like that? Did you see anything like that? What did you use to calibrate, I would say?
The calibrate, you mean the calibration process, right, Christy?
Yeah, I mean, we all use that term differently, but I would say like, yeah, in your Ecosim model, did you use FWC data?
Well, I don't tune the Ecosim model. So what I did is just believe that a person who know the system, as my former advisor, acknowledged that probably the most feasible representation of the system was the Ecosim predictions. So we believe that our Ecosim predictions were representing the true state of the system. So
But
what
did
we
you
did,
use time series from
yeah,
what from FWC?
well, we didn't compare with the time series. The time series we're using for fitting the Ecosim, but the comparison with the see, yeah, the fitting of the Ecosim was done with different. I don't recall, but there were from the CMAP trolling data. There are a bunch of fisher independent
Yeah.
data there.
Okay. Okay. Yeah, that's my question. I just wanted that clarity to make sure that.
Yeah, there are also bait fish data, a bunch of surveys. The ones that also we use to were the ones that we use also to evaluate those, well, validate those red tide effects later.
Do you have a follow-on question, Christy, or can Carl go?
No, all you, Carl.
Just to continue that, we tried in the initial WFS model fitting to use some of the FIM data, the fishery independent monitoring data, and we had a lot of trouble with it. And I think, incorrectly didn't use the FIM data that showed the pulse that Daniel showed there in small creatures going much higher than the model predicted. If we had been fitting with those data, we might have captured some of those those trophic responses by little things like crabs and mullets and that the commercial fishermen target near shore.
The FIM data is the one that is shallow, right?
Yeah, it's all right close to
For
shore.
small
and
pelagics.
It's a variety of gears, and it doesn't sample some things very well. Mullets
Yeah.
jump out of the nets, so nobody trusts the data and crazy things like that. So we kind of gave up on using it. Which is a
Yeah,
real
we
shame.
use that data to evaluate the effects of red tide, but I don't know if you see on the presentation, there is this small polygon in the central region that probably we didn't have much an overview of what is happening in the whole ecosystem. That
Yeah.
Yeah. Okay, any more questions from anybody online or anybody? Oh, sorry, Kim, you were yellow and green there. So.
Yeah, great talk, Daniel. I had a follow-up question on the fitting as well. So you ended up using your Ecosim outputs kind of as your true predictions, and then looked how close your Ecospace output was to that. And you know, with this spatial modeling, I bet you really see the value in these, you know, spatial features that actually add information to how your biomass changes over time, right? So I'm wondering, by doing that, aren't you specifically selecting the model with the lowest sensibility to spatial stressors, just because then it would mostly resemble your Ecosim? So you're kind of trying to really reduce the effect of what Ecospace adds when you do your fitting like that.
Yeah, if you see from that point of view, yeah, it could be, but you can think on the other way, like, well, we are getting those predictions from Ecosim, but they also include that spatial temporal information, even we are not representing that. And I lost the track of the question. Sorry, Kim.
Oh, I was just wondering, by taking Ecosim as your true predictions, aren't you, and using root mean square error to fit your Ecospace to that, aren't you reducing what you add spatially as much as possible to get the best fit?
Well, depends on how you see that, because also we have other when you incorporate that spatial temporal information, you have many outcomes depends on the information that you are inputting and depending on the species. Even there are some species that may increase exponentially, as we see, or may decrease, but we kind of were looking for, not, I wouldn't say removing that or trying to lower the importance of spatial temporal data, but I would say that we want to approximate, even we are getting information, additional information, trying to not getting like unfeasible predictions.
Right. Yeah, no, I indeed, like taking out like exponential growth and crashes is a great thing to look at. For fitting.
Maybe using observation will be more valuable. I don't know. This is something that may be worth to check, but I will have the same questions as if I'm using the Ecosim. Maybe I'm using some predictions to fit, but if I'm using observation, maybe I'm using observation that are not capturing the red tide effects. So
Right.
it's
Yeah,
kind of tricky.
I'm wondering if you're like on your slide 25 that your best root mean square error comes from your model with the lowest sensitivity, whether that's related to that, to how you fit, but I'm not sure.
Would you say the 25?
Like slide 25, you have like your low sensitivity model, I think is your best or your lowest root mean square error.
Yeah, it was kind of different between the evaluating the how we approximate to the Ecosim and how we approach the natural mortality. Yeah.
Okay, maybe you guys can have a conversation about this.
Yeah, this is
Yeah.
more maybe a discussion.
yeah,
But
yeah.
yeah, I really like it.
We have a question
Yeah.
from Studio. I don't know if you want to put your camera on so we can see you as well. And there we go. Welcome.
Thank you to give me this opportunity to ask, and thank you, Daniel, for the great talk. I have two questions. The first one about the input data in your model, and my second question, if this model, we can apply this model to assess the plankton biomass, especially every time we have a direct relationship between small pelagic fish species and plankton biomass.
And the first question was related with the input data, you said, Sidi?
Yeah, because I lost the beginning of your talk and just like
No worries. It was so for our Ecospace model, we use information on different environmental variables for like a spatial temporal. Well, it was raster data, but and we also have apart from that environmental data, we have the response functions of each functional group to each environmental factor in the model. If I remember well, was that. And what else? You asked about if we can apply that to phytoplankton, you said?
Yes.
I mean, you can represent phytoplankton as a primary production in Ecospace, but in our case, because the implication of that phytoplankton was with this toxicity, we wanted to use it as an environmental factor. So there are different ways of implementing one thing and the other one.
This is great. Why ask? I work.
I think he's gone.
That's a trick question.
Yeah, I think he was being, it's us frozen. Oh, well, maybe they can speak to each other then. Have we run out of?
Also, especially according to the acoustic data, we give the allocation for the small pelagic fish species, and if we don't have a special survey for the plankton, especially we don't give any importance for the plankton biomass. And the things I want to use this data, and if it's possible to combine the between acoustic data and use also Ecopath model to assess that.
Well, you can incorporate the distribution of a species in. I think that instead of using response functions, I think that you can drive the model using distributions. So you can try the other one.
Well, thank you. I will. I request to keep touch with you and your supervisor to have more advice in this context. And thank you for giving me this.
Sure. Thank you. You're welcome.
Thank you very much.
Excellent. Do we have any other questions in the room? Any other questions online? I think we're over our time anyway. So if there's no other questions, thank you very much, Daniel. That was great. It was nice.
Thank you, Sheila, for giving me that opportunity.